Hello everybody. Today I'll be doing the iodine and starch test. This is the iodine I'll be using, so let's get started. The purpose of this experiment is to use iodine to test for starch in different foods. When iodine and starch are mixed together, the normally white starch and the normally yellow-orange iodine combine to form a blue-black complex. Starch is made of amylose and amylopectin, two different polysaccharides. Amylose is a linear helical compound. Amylopectin is a highly branched polysaccharide. This is important because although the exact mechanism of this reaction is unknown, it is believed that iodine 5 ions become trapped in the coils of the helical amylose. This changes the light absorbing properties of the mixture, and so when light comes in, most of it is absorbed, only letting out the deepest, darkest blue light. So if you want to test to see if a food has starch in it, you can add a drop of iodine to see if it changes into that deep dark or blue color. So to begin our experiment, we first need to test our controls. We have two controls, a negative and a positive control. Our negative control is just plain water and should elicit no response. Our positive control is a water and starch mixture, which should elicit a positive response. We have the iodine in this bowl to make it easier to pour, so we will test our iodine now. We add some iodine to the plain water and we see that it stays its normal yellow-orange color. When we add iodine to the starch and water mixture, we see that it immediately changes into a deep blue, almost even a black color. So this iodine passed both controls and had no response with the plain water and had a positive response when mixed with pure starch. So now for our samples, we have 8 different samples. We have sugar, bread, an orange slice, olive oil, oats, ham, boiled potato, and onion. Some of these compounds were chosen because I had a feeling there would be a response. Some were chosen because I thought there would be no response whatsoever. And some I'm not really sure what to expect. So let's get started. We're gonna test the sugar first. And you can see that the sugar did not change into a dark color. It pretty much stayed the normal amber color that iodine is. We'll do the bread now. So you can see in contrast that the bread turned a very dark color. So the bread is positive for starch while the sugar isn't. We'll move to the orange now. So the orange also did not change color, so there is no starch in the orange slice. And finally, olive oil. The olive oil also did not elicit a response. If you look closely, the iodine still appears to be its amber color. Move over to the second plate. We'll start with the oats. So if you look closely at the oats, you can see that the iodine did in fact change into a very dark color. The ham did not change color. You can tell that the iodine is still amber. Now potato. The potato certainly elicited a positive result. You can see how black it is. And finally, onion. The onion appears to not elicit a response as the iodine still appears amber. So looking at our 8 samples, most of these results were what I expected them to be. Sugar and orange slice are both composed of simple carbohydrates, so they should have elicited no response, which is exactly what we got. Olive oil is a fat and contains no carbohydrates, so there should be no response from that either. Ham is a protein and not a carbohydrate, so there should be no positive result from the ham. And finally, the bread, the oats, and the potato all elicited positive results, which I think was to be expected, as most people kind of intuitively know that those 3 foods contain starch in them. The one I was most surprised by was the onion, because I did kind of expect the onion to react with the iodine, but I guess the onion doesn't have that much starch in it to be able to react with the iodine. So thank you for tuning into this experiment, and have a good day.